Okay, welcome everyone to the beginning of enzymology lessons. This is going to be lesson one, and I'm Jellyverse as usual. So let me change my pointer to a laser pointer so that you start on with the lessons. So these slides, I'll be sharing it later on after lesson one. It contains some of the information that may not be mentioned in your lectures, so I urge you to pay close attention to this as we move on. I'll be taking my time from the beginning of the course. So this is lesson one and it entails the beginning of everything up to the start of classification of enzymes. Okay, so let's start with something called the coupled reaction or the thermodynamic consideration. So when you're talking about coupled reactions in cells, remember some of the reactions are there in the cells. They can occur naturally, they occur spontaneously. They don't need anything to push it, or they don't need anything to drive it for it to occur. Others too are there. They need to be coupled with other reactions in order for them to occur. For reactions that occur naturally, they are having a gift-free energy to be negative. That is ideally reactions that occur spontaneously in cells are negatively gift-free energy. However, some of the biological reactions, such as protein synthesis, where you are having amino acid forming the peptide bond in order for protein to be formed, it is non-spontaneous. It requires the energy input. That's why we talk about the hydrolysis of ATP. Because when you hydrolyze ATP, it releases an energy, and this energy will act as a compensation. So as we move on, we are going to understand whatever that we are doing here. So I've discussed everything here. I think from the first line to the fourth line, I've discussed it. Right. So there are some conditions for a coupled reaction. For you to couple a reaction in a cell or maybe in a test tube, one, the sum of their gives-free energy for the two reactions that you are coupling, it should be negative in order for the reaction to occur or in order for the reaction to be spontaneous. Then we are going to look at some of the examples why I'm saying that after the summation of these two reactions, your gate-free energy should be negative. And also, since we are adding two reactions, we expect that after writing the general equation, we should have their common intermediate cancelling out, meaning a common intermediate should be on both sides so that you can cancel out based on the rules of summing up equations, which was being treated in thermodynamics, I think Chem 217. Okay, so let's move on. Example of the coupled reaction is what you are seeing here. ATP hydrolysis. When you couple ATP hydrolysis is an energy releasing. It releases energy. So look at it. If you're having ATP and you react it with water, it's going to hydrolyze to give you ADP. ATP in BCME 201, we talked about it as what? As a source of energy. So it hydrolyzes to give ADP and inorganic pi. ATP is adenosine triphosphate, meaning adenine that has been bonded to its nitrogenous base plus your sugar and also your triphosphate, which is 3-phosphate. In the presence of water, it's going to hydrolyze, and one of the phosphate group is going to leave as the Pi, which is the inorganic phosphate. The Pi here is called inorganic phosphate. So with this, if this reaction should occur, your gift-free energy is negative 30.5 kilojoules, meaning the reaction is spontaneous. The reaction can occur on its own without adding anything to it. On the other side, which is step 2, in step 2, which is phosphorylation of glucose, meaning we are adding a phosphate group to glucose. So in this particular first reaction, we released a Pi, which is inorganic phosphate. In the second reaction, we are adding the inorganic phosphate to the glucose in order to get glucose 6-phosphate plus water. Now there's something I want you to know here. This reaction is endothermic. It requires energy. Look at the gate-free energy. The gate-free energy is positive. So positive 13.8. The moment you are having your gate-free energy to be a positive value, meaning the reaction needs to be driven by other reactions. It is not spontaneous. So when we are adding these two reactions together, negative 30.5 plus positive 13.8, your overall gift-free energy or your change in gift-free energy is negative 16.7, meaning the reaction is spontaneous now. Meaning you can add a phosphate group to glucose and the reaction will proceed because you have coupled it with ATP hydrolysis. And remember, if you add these two, you are going to have your inorganic phosphate. Remember, if you are adding these two, this and this, let me try and use a pen to illustrate it to you. So if you are adding this particular equation to this, remember, this is an intermediate on the right side, but this is an intermediate on the left side. So Pi and Pi will cancel out. And when they cancel out, you are going to get your overall equation to be ATP plus your glucose, giving you ADP, as you can see here, plus your glucose 6-phosphate. You see how simple it is, but generally, this is what is occurring. You added these two reactions in order to get a full coupled reaction or overall coupled reaction. So you have to note the overall gift-free energy is negative. So the coupled reaction will proceed spontaneously. The Pi that you are seeing here, which is the pi acts as a common intermediate. The pi acts as a common intermediate. But water is generally not a common intermediate, and I'm going to give you the reason why water is not a common intermediate in the next slide. So let's move on to the next slide. So in the next slide, look at another reaction too. But in the next slide, this is the reason why water is not part. So even though or although the reactions share a common intermediate, the total gift-free energy is positive. But when you are having a situation where after coupling the reaction, still your overall gift-free energy is still positive, then this means that your reaction is not spontaneous. For instance, the reaction that you are coupling it with, in order for you to make the reaction spontaneous, you are having it to be having a lesser value, a lesser negative value as compared to the positive value of the reaction that is not spontaneous. This means obviously you are not going to have your reaction occurring. This is what I mean. If you are having a negative value for the reaction that you are using to couple it with, it should be greater than 15. In fact, it should be greater than 16 for the reaction to spontaneously occur. But if it is lesser than 15.9, obviously, even if you add it up, you are still going to get a positive value, which is saying that the reaction will still be non-spontaneous. So this reaction is a typical example of the reactions in which we are coupling the reaction, but still the reaction is not occurring. This is what I mean. The first equation, where we are having your ATP plus your H2O, and this is what you're having. After adding all of them, you see you're having your total gift-free energy to be negative 16.7, meaning the reaction is feasible, the reaction is spontaneous. But let's look at the second example that we talked about. You're having glycerol 1-phosphate hydrolyzing with water to give you glycerol and Pi. At the same time, we are adding inorganic phosphate to fructose to give you fructose 6-phosphate. And the energy that is required is 15.9. Here, the energy that is released is 9.9, negative 9.9. This means that this energy is still not enough to compensate this amount of energy required to make the addition of inorganic phosphate to fructose to give you fructose 6-phosphate feasible. There is no enough energy. You should get around 16 and above so that when you add it, you are going to get a negative value. The moment you're having a negative value, it means that reaction is spontaneous. That reaction can occur easily. So that explains what I've said earlier. Now let's look at the reason why water is not a common intermediate. Water is not a common intermediate in the coupled reaction because it is present in a high concentration in water cells. Remember, in a cell, in a particular cell, for instance, in a human cell, in an animal cell, in a plant cell, the amount of water or the content of water in each cell is very high. So it is having a very little free energy to transfer. And for that matter, it serves mainly as a stable byproduct. or simply it acts as a solvent rather than an energy carrier. If you're able to explain with this particular phrase there, you are good to go. Why water is not a common intermediate? Water is not a common intermediate because its concentration in cells is very high, and also it is stable as well. So it's a stable byproduct, and it acts as a solvent for the reaction to occur rather than acting as an energy carrier. It doesn't carry energy. You get it? So that is the explanation why water is not a common intermediate. You can't cancel out water. Even though the reaction, we are having water here and you're having water. You're having water on this side. You are also having water on this side. You can still not cancel it out. It needs to be there. The reason why it needs to be there is that water acts as a solvent for the reaction to occur rather than acting as an energy carrier. So let's move on to the next slide. We are talking about the standard Gibbs free energy. So the standard Gibbs free energy is a difference between the energy, right? It's the difference in free energy between the reactant and the product of a reaction under the standard conditions. What are the standard conditions? The standard conditions here from what we learned in Chem 217 thermodynamics is that your pressure is 1 atm or 1 atmospheric pressure. Your concentration is 1 mole per dm cube, and your temperature is 25 degrees Celsius, usually 25 degrees Celsius, which is 298 Kelvin, and your pH is 0. Now let's look at something here. In a human system or in biological system, you can have pressure 1 constant. You can have a concentration of, let's say, the human blood or concentration of a particular substance in the cell to be stationary 1 mole per dm cube. No. And likewise, you can have the temperature. Homeostasis will not allow your temperature to be 25 degrees Celsius constantly. And the last one is pH. Remember, our pH, for which most of our enzymes work, is around 7. So how come we are going to have pH to be equal to 0? So this is that we have to introduce something called the Gibbs free energy delta. That's the one that compensates for all the conditions that is occurring in the biological system. So the Gibbs free energy, the values are commonly listed in scientific literature, right? It's true, the values are listed in scientific literature. If you have to be moving back to the previous slide where we are having some specific values for each of the reactions. Now, they are useful in predicting whether a reaction is likely to occur or not. That is the purpose of the Gibbs free energy. Now, the limitations of the Gibbs free energy in living cells, I've already said it earlier. The chemical reactions inside the living cells do not occur under standard conditions. For example, the pH inside the living cell is about 7. It's not 0. Like I said earlier, the concentration of the substance constantly changes. So that one too is also another point of which acts as a limitation for the Gibbs free energy. And the last one is that the cellular conditions are highly dynamic. They changes a lot. You can't have it being stagnant like how the standard conditions are saying, as read earlier. So because of this, biochemists usually do not rely on this one alone. They don't rely on the standard Gibbs free energy alone. They rather rely on the delta as well. So that's what we are moving to. So Gibbs free energy under cellular condition, which is the Gibbs free energy delta. So the actual free energy change under non-standard cellular conditions is what we are referring to as the Gibbs free energy delta. So now let's look at something. The importance is that, and this is a typical interpretation. If you are having your gene to be negative, that's what you are seeing here. If you're having your gene to be negative, if your gene is negative for the first one, you are going to have your reaction to be spontaneous. It means your reaction will proceed forward. If your gene is positive, which is the second option, you are going to have the reaction to be non-spontaneous. It means you require another option. So you have to couple it with another reaction in order for it to occur. And we know that if your gene is equal to 0 or your change in Gibbs free energy is equal to 0, then obviously you should note that your reaction is at equilibrium. Molecularly, the reaction is occurring, but physically, the reaction is not moving forward or backward. It's at equilibrium. So let's go and talk about the delta G prime. Now for the delta J prime, if the pH is actually fixed at 7, unlike how for the standard Gibbs free energy you are having your pH to be equal to 0, in the delta prime you are having your pH to be fixed at 7, and pH of 7 is actually a physiological pH where most of the cells or most of the enzymes can function properly. Temperature and pressure remains at the standard values, like 27 degrees Celsius. The concentrations of some of the substances other than protons are also kept under the standard conditions, like we said. So this makes this particular delta J prime more suitable for biological systems. The reason is that for delta J prime, it follows the principle of making it having a pH of 7, and that is the pH at which most of the cells work or most of the cells function. Okay, or most of the enzymes also work well. Now the value of the gene, it depends on the temperature. The value of the Gibbs free energy, it depends on the temperature and also the concentration of the substances. So because of these conditions here, we are having exceptions. For a same reaction, you can have a same reaction where we are considering it to be, let's say, feasible, or the reaction is spontaneous. Under a different condition with the same reaction, you are going to have the reaction not occurring because you have changed the conditions. If you are increasing the concentration of the reactant, it is likely possible that the reaction that we consider it to be feasible or spontaneous will be likely non-spontaneous. We are going to look at some of the reasons why these things happen. So now the change in free energy under the non-standard conditions can be calculated using this equation where we are having our change in J to be equal to our standard Gibbs free energy plus our R. Our R is the gas constant, our temperature, which is the T, and our natural logarithm. This is the concentration of the products, and this is the concentration of the reactants. Now, this equation shows that the free Gibbs free energy depends on the temperature. Remember, we are having T here. It depends on the temperature. It also depends on the concentration of both the products and the reactants, like what you are seeing here. So because of the cellular reactions, all these things are constant. Because of this, you can have your Gibbs free energy changing, or your changing Gibbs free energy to be changing or being different from this. Okay, let's move on. So when you are having your concentration of your product and your concentration of your reactants being the same, remember the whole of this goes to 0. And then we are going to have a situation where our change in Gibbs free energy is the same thing as our change in standard Gibbs free energy, meaning the reaction is at equilibrium or the process is at equilibrium. So this is the Gibbs free energy, the free energy change under the actual condition that we are considering. This is the standard free energy change. This is the gas constant. This is the absolute temperature in Kelvin. And this is the natural logarithm, which is a ln. Right. So now when the product of the concentration increases, when we are having the product increasing, right, we are going to have your gene to be more positive. And what happens when we are having our gene to be more positive? When our gene is more positive, we are going to have the reaction to be less spontaneous. The reaction will not be occurring. That's why I was saying that for a same reaction, let's say we are having A plus B, right? A plus B giving you C. If the reaction should be proceeding spontaneously, and by chance you are increasing the concentration of the product, we are going to make this change in Gibbs free energy for the actual condition. We are going to make it become more positive. And if it is more positive, what do you think it will happen? The greater the value in terms of positive, we are going to have the reaction to be less spontaneous, or the reaction is not going to occur. When the reactant concentration also increases, then you are going to have the ratio to become a very smaller value, and then your gene will be more negative. So for that matter, we are going to have our reaction to be increasing. So they can ask you a question. We'll be solving a lot of questions where we'll be introduced into some of the questions that it will let you understand the course very well. For instance, if you are increasing the product, what will happen to the gift-free energy? Will the reaction be more spontaneous or it will be less spontaneous? That one will also count. If you are also increasing the concentration of the reactant, what do you think it will happen to the reaction? Will the reaction will the reaction be feasible or it will be less feasible? And I've said it here that if you increase the concentration of the products, your reaction will be less spontaneous. Your reaction will be less spontaneous. It will not be driven forward. But if you are increasing the concentration of the reactants, because your ratio will become smaller, and you know mathematics, if you are having this equation, right, and your ratio is smaller, you are going to have a smaller value. But if your product is increasing, if the concentration of the product is increasing, then this means that you're actually going to have a bigger value. And this bigger value is actually going to affect the value. It's going to become more positive. And when this becomes more positive, you know what will happen. The reaction will be less spontaneous. Now let's note. So even if gene, when we are having a condition where our change in gene is greater than 0, a reaction can still proceed spontaneously. That is when you increase the concentration of the reactant. So you could see that I've given a caution here that a reaction can still proceed spontaneously in cells if the concentration of the reactants and the products make this particular value negative. So it depends on the concentration, like we said, and also depends on the temperature. It also depends on the gas constant. The gas constant is actually there. It's a constant value, so we can decide not to include it among the answers. But when we are looking at the main point, it depends on the concentration. It depends on the products, and it also depends on the reactants. These are the main three things that we need. Don't forget, these are the main three things. So you can jot it down somewhere. Temperature, concentration of the reactant, and products. Okay, so now let's move on. So this is why many biochemical reactions occurring in living cells, even though their gift-free energy values suggest that they should not proceed. Some of the reactions are there. If you look at it in the biological system, you could see that no, this reaction is not supposed to occur. But it is occurring. It is occurring because of these conditions that we are having greater amount of reactants, right? And some of the reactions too are there. We assume that it should not occur, or it should occur rather. It should be spontaneous, but it is not occurring because we are having the products being greater. The concentration of the products, we're having a lot of products being accumulated. That reaction will not occur. Okay, so let's move on. Now, this actually represents energy diagrams where we are having our gift-free energy. So this particular reaction shows our reaction where we are having our gift-free energy to be positive value. Here we are having a positive value, and this side we are having a negative value. So it's the product minus the reactant, and we are having a positive value because this is the energy. This is the energy level. If you're having your reactant here, let's say if your reactant is 2, if your energy of your reactant is 2, you are going to have your energy of your product to be around 4, because this is where the product is. This is where the product is, and this is where your reactants are. So product minus reactant, 4 minus 2 will give you what? Positive 2. It's a positive value. But with this, look at where our reactant is. Our reactant, let's say if our reactant is 4, see where our product is. Our product might be around 2. Obviously, we are going to have 2 minus 4, which is negative 2. This reaction is spontaneous, but this reaction is not spontaneous. So let's look at it here. So A is non-spontaneous, but B is a spontaneous reaction. Now let's look at this particular concept here. It's very important. If you are saying a reaction is spontaneous, does that mean that the reaction will be fast or it will be slow? Now there's one thing I want to say before we move on to it. The spontaneity of a reaction does not determine whether a reaction will be fast or it will be slow. So in terms of thermodynamics and chemical kinetics, thermodynamics considerations will help us determine whether a reaction is favorable, like I said, whether the reaction is actually going to proceed forward or proceed backward. That is the function of the thermodynamics. For the reaction to be faster or slower, it depends on the kinetics of the reaction. It depends on the molecules. It depends on the temperature. It depends on how colliding the molecules are in their right orientation. So, however, thermodynamics does not tell the pathway a reaction follows, does not also tell us how fast the reaction occurs. A reaction may be spontaneous, but yet you are going to have it occurring very slow. That is it. Because kinetics is not related to thermodynamics. For a reaction to occur or not does not correlate with how the reaction is going to be, whether it is fast or slow. Now, let's look at a reaction here. This reaction is actually feasible. If you're having sucrose, example of sucrose is, let's say, sugar. When you leave sugar there, oxidation will occur. So oxidation of sugar is a feasible reaction in the presence of air or oxygen. You are going to have your sugar being oxidized to give you carbon dioxide and 11 moles of water. Now let's look at something here. When you are having sugar in real life, when you put sugar on a table and expose it to air, do you think something happens to the sugar? There is no noticeable change that happens to the sugar. The sugar can be there on a table. Let's say this is our table. You are going to have your sugar being on the table for a very longer time. Nothing will happen to the sugar. The sugar will not be converted to carbon dioxide. You are not going to see it. Meanwhile, for the sugar to be converted to carbon dioxide, we are seeing that the reaction is feasible. Then this means that maybe after 1 million years, maybe you are going to have your sugar being converted to carbon dioxide. Who knows? But with our chemistry that we are learning here, we are seeing that the reaction is feasible because it is having a negative value, and negative gives free energy. So since the gives free energy change is negative, the reaction is thermodynamically favorable, right? It should occur spontaneously. It should occur without coupling it with other reactions. But the observation is that you are not going to have the sugar being converted to carbon dioxide. It will be there for a very longer time. That is to tell you that even though a reaction is spontaneous, it does not tell you whether the reaction will be fast or slow. So now the reason is that the reaction occurs extremely slow at room temperature. That's why I was saying that maybe it is occurring, but it's occurring very, very, very slow. So you can't see a noticeable change. So it tells the oxidation of sucrose is thermodynamically favorable, but kinetically it is stable. It is very slow. If you are saying something is kinetically stable, you are not going to see the reaction actually occurring, even though it might be occurring in the molecular level. But if it is kinetically unstable, you are going to have the reaction occurring very fast. Okay, now let's look at the hydrolysis of ATP. It's also very important in our lesson here. We need to focus on that as well. Hydrolysis of ATP. Now, if you're having your ATP in the presence of water, it hydrolyzes to give you ADP, which is adenosine diphosphate, plus your inorganic pi or your inorganic phosphate. Remember, the thing is inorganic phosphate, not inorganic pi, like I've been saying. So always make sure you put it in the right way. It's inorganic phosphate. So this reaction is spontaneous. In a test tube at 37 degrees Celsius and a pH of 7, the reaction will take about 4 weeks to occur. Why? Yes, the reaction, even though we are having a positive value, in a test tube, the reaction will take about 4 weeks for this to complete, for us to get our inorganic phosphate. But inside the living cells, if you want to move your hand, if you want to grab something, you require energy. This energy comes from hydrolysis of ATP. So assuming what's happening in the test tube is the same thing that is happening in real life, in our living cells. This means that, let's say, if you want to slap someone, it will take you 4 weeks for you to slap the person. You get it? Inside the living cells, that is not it. It will take few seconds. If you want to give someone a knock and you are ready to give the person a knock, it won't even take you a second. You will just give the person the knock instantly. That is different from what's occurring in the test tube. The reason why the reaction is faster is because there's a presence of enzyme. Enzyme is there. Enzymes are in living cells. So living cells contain enzymes that speed up the reaction. In test tubes, you're not having enzyme, but assuming in the test tube you add the enzyme hydrolase. This enzyme can make the conversion of ATP to ADP in a very few seconds. It can make it occur in a very few seconds. Yes, because there's a presence of the enzyme there. What do enzymes do for this particular reaction to be faster? The enzyme will lower the activation energy. It will also increase the reaction rate. The moment you are having your activation energy being lowered, your reaction rate will be increased. And also, it does not change the Gibbs-free energy, or it does not change the spontaneity of the reaction. For instance, if a particular reaction is set to proceed forward, when you add enzymes to it, that enzyme is not going to change it from being spontaneous to non-spontaneous. No. Enzymes just speed up reactions. That is why when you are having an equilibrium reaction, if you add enzymes to it, it doesn't have any effect on it. This is a typical example. Now, without enzymes, many of the essential biochemical reactions would occur very slow. And you think like we will be able to survive life? Let's say you want to kick something and now you don't have enzymes. There are no enzymes in your system. You want to open your mouth and eat. Open your mouth, you require energy. And you have to take 4 weeks before you open your mouth to eat. No. Enzymes are very important. That's why we have enzymes in our living systems, in our living cells. Without enzymes, human being cannot survive. So, for all what I've said, in terms of thermodynamics and kinetics, the thermodynamics will tell us whether a reaction can occur. But for the kinetics, it tells us how fast or slow the reaction will be. Thermodynamics, it depends on the value of G. That's why when you're having a positive value, we are saying that the reaction is non-spontaneous. If you're having a negative value, we are saying that the reaction is spontaneous. But in terms of kinetics, it depends on the activation energy. It depends only on the activation energy. For instance, let me move back to the previous slides. This is the activation energy. This is your activation energy. If your activation energy is very high, you are going to have your reaction being slower because it will take time for you to cross this energy barrier where you are going to form your transition compound. This is a transition state. This is where you are going to form your transient molecule. The transient molecule is very unstable. Compounds that are having a higher energy are very unstable. Look at where the value of this transient molecule is. It's around this side. It's having a higher amount of energy. It's very unstable. So for the amount of energy that it will require for your molecule to reach this stage, it will make the reaction very slow. So the lower the activation energy, then the faster the reaction. The higher the activation energy, the slower the reaction. Like I've said earlier. So I've said this thing too. Faster reactions will lead to lower activation energy. Now, a spontaneous reaction does not always occur rapidly, like I've been saying always. It depends on two things. For the reaction to occur, it depends on the thermodynamic aspect or the concept. For the reaction to occur fast, it occurs based on the kinetics, whether the activation energy is higher or slow. So for a reaction to be proceeding very faster, you are going to have your enzyme. That's why we need our enzyme. So if the energy barrier, let's say if you are having your reaction energy profile and your energy barrier to make your reaction occur, if this is the energy barrier, right? In the presence of enzymes also like a catalyst, but enzymes are biological catalysts. You get to enzymes when you're talking about it very well, you understand it very well. Enzymes are found in living systems, yes. What the enzyme will do is that it will create a new reaction pathway. It will reduce the energy barrier. So here now you are going to have this to be your energy barrier, a very small or short one. And what did we say? If you are having a lower activation energy, you are going to have your reaction proceeding faster. That's why when you're having your enzymes in the system, you are going to have your reaction producing or your reaction proceeding very faster as compared to the reaction without the enzymes. For instance, 4 weeks and few seconds. Enzymes will convert this large period, 4 weeks, to just few seconds. Okay, so let's move on. Methods, right? Now let's look at the methods. Living cells require reactions, right, to occur rapidly. Living cells, they require reactions to occur rapidly, and it is enough to support life. So the two major methods of increasing the reaction rate is increasing the temperature. These two things, never forget about it. For a reaction to proceed rapidly enough to support life, the two things that we need is increase in temperature and catalysis. But remember, the temperature also comes with some it comes with a problem because catalysis we need enzymes, right? Enzymes are proteins. So if you're having increase in temperature, if the temperature is very high, what happens to proteins when the temperature is very high or they are in a higher temperature environment? They denature, so the enzyme is going to lose its activity. So an increase in temperature in laboratory, for instance, your chemical reactions will often speed up when you heat the reaction. It is true. The reason is that one, the molecules are going to move faster. Collision is also going to become frequent, and your collisions will lead to the faster reactions. If you're having frequent collisions, the rate at which they are going to collide in the correct orientation will also be very high. For instance, if you are having a wall and your molecules are supposed to collide in this direction alone, if the collision or if the molecules are not moving faster, this means that if it will take 2 seconds for the molecule to hit it and you increase the temperature, it will take it like only few seconds, like microsecond. It's taking like 0.5 microsecond for it to collide. So the more it is colliding, the chances of it colliding the right orientation will be very high. So your reaction will also be what? Will be faster. Now let's move on. The limitation in the living cell is that even in the living cells, when you are sick and your temperature is very high, they tend to cool it down. So high temperatures can damage cells, and the proteins too may also denature. If there's a presence of enzymes there, your proteins are going to be denatured or your enzymes are going to be denatured, meaning the enzymes are going to lose its activity, and the probability that these enzymes will not regain their function is very high because the moment you denature an enzyme, it's going to lose its function mostly. Now, the cells will use another method in order to accelerate its reaction. If high temperature is not helping because it is damaging the cells, then we need a different alternative. That's when we introduce the enzymes, and the enzyme is related to the catalysis. So many reactions can be accelerated by substances called the catalyst. Catalysts will include metal ions, it can include acid, it can include bases. All these things are inorganic what catalysts. The organic catalysts are what we are referring to as the enzymes. So for example, the hydrolysis of ATP occurs faster when there's small amount of acid. If you are having ATP, this same reaction, if there is no enzyme and you are increasing the temperature, the rate at which you are going to have your reaction occurring will be faster. So if it will take you 4 weeks for the reaction to complete without any catalyst or any enzyme, if you increase the temperature, it's likely you are going to use just a day in order to do it, or a few seconds for it to happen, as you can see. Also, if you're having enzyme, the same thing will also happen. So for the biological catalysts, they are the ones that are found in the living cells. So living cells produce highly efficient catalysts called the enzymes. These enzymes are much effective than the original inorganic catalysts, like I said. The reason why they are effective is that they are very specific. If they are meant to make a particular reaction occur, let's say if they are meant to convert W to Z, that is the only function that that particular enzyme can do. But look at inorganic catalysts like H2SO4. H2SO4 is capable of making a lot of reactions be driven faster, different reactions. So an advantage of the biological catalyst or the enzymes is that enzymes are very specific in their reactions. So thousands of enzymes exist in cells, and each enzyme usually catalyzes one specific reactions. So nearly all reactions in the living organisms depend on enzymes, right? Every reaction that's occurring in our living system now, it depends on enzyme. There's a particular enzyme that is making it possible. Without the enzyme, then life will not be existing at all in the first place because everything will be moving very slowly. So enzyme can produce extremely large increase in temperature rates. Example, let's look at the enzyme urease. Urease is an enzyme that is capable of hydrolyzing urea about 10 raised to the power 14 times faster than when we are not having any urease in the solution. So if you are having a solution containing urea and you want to hydrolyze this urea, if you don't add any enzyme, if there is no urease enzyme in there, it can take you like, let's say, 1 million years. But if you add the urease into it, if you add the enzyme urease into it, then this means that we are capable of what? Minimizing the duration that it will occur, or the duration that it will take for the reaction to occur. You can minimize it by 10 raised to the power 14 times. So the reaction will be 10 raised to the power 14 times faster when there's catalyst than when there is no catalyst. So in a very small amount, the enzymes are very effective. If you add a very small enzyme, small amount of enzyme, still the reaction will occur, and it's going to make the reaction 10 raised to the power 14 times faster. Now let's move on. So enzymes and their thermodynamics. Enzymes speed up reactions that are thermodynamically feasible. Like I said, if you're having a reaction and the reaction is non-spontaneous, no matter the amount of enzyme that you add, the enzyme cannot make the reaction spontaneous. Keep it in mind. If your reaction cannot occur under normal conditions, if you add enzyme to it, the enzyme is not going to make the reaction occur. An enzyme does not, this is because an enzyme does not change your Gibbs-free energy. It does not change it from positive to negative. The enzyme does not also change equilibrium. If a reaction is at equilibrium, the enzyme is not going to change the reaction from being equilibrium to, let's say, forward reaction. And also, it does not affect the spontaneity of the reaction. The only thing that the enzyme can do is to lower the activation energy. I want you to understand this concept very well because when we are talking about the activities of enzyme, this thing will be required. Now, the specificity of enzymes. Enzymes are usually specific. Each enzyme acts on a particular substrate and catalyzes a specific reaction. So they generally work in this particular temperature range. They work from 10 degrees Celsius to 50 degrees Celsius. And usually they work at a pH around 7, a neutral pH of 7. That's where they work. So now let's look at enzyme, how an enzyme looks like. This is an enzyme. Enzyme is a protein, right? And it falls under the tertiary level. This is the catalytic site, and this is the substrate binding site. When we are looking at enzyme, enzyme is just a protein, right? And at the tertiary level, but it is having a cleft or it's having a pocket. This pocket is where we are going to have your substrate coming to bind to. So it binds to that substrate. Let me put it in this way. The enzyme binds to the substrate. When it binds to the substrate, it needs to grip it. What grips it is the substrate binding site. Remember, all what you are using is sites. You can see sites there. This is that it's a place in the enzyme. And proteins mix up the enzyme. What are proteins made up of? They are made up of amino acid. This means that in this catalytic site or the substrate binding site that you are talking about, you are going to find amino acids in there. So amino acids are rather found in this site, in this catalytic site or in this substrate binding site. So when we are moving on, I'll let you understand the main reason why we are having these groups, the catalytic sites and binding sites, and their importance, what we actually need to know about it. So unlike enzymes, ordinary chemical catalysts can function under extreme conditions such as a very high temperature, a very high pressure, a strong acidic or alkaline conditions. Enzyme cannot do that. Ordinary catalysts like A2SO4, they can all perform their functions under these conditions, but enzyme cannot do that. Enzymes will be denatured. So the advantages of the enzymes here is that a major advantage of the enzyme over this chemical catalyst is that it is precise, right? The precise arrangement of the catalytic groups. So even though you can have an enzyme, so when we are moving forward, we are going to realize that you can have an enzyme all right if the shape of the enzyme active site is this, and you are having a substrate. If this is a substrate, this substrate can fit into it, right? Don't mind my drawing, but I'm just trying to show you something here. Look at the shape of the active site and look at the substrate. This can fit into the active site, but there is specificity in the enzyme. If the arrangement of the catalytic groups are not correct to match with the substrate, you can have your substrate binding to the active site all right, but no reaction will occur. That is one advantage of what enzymes. Enzymes are very specific. So until there's a proper binding of the substrate to the active site, there is no way catalysis will occur, or there is no way your reaction will occur. It can actually bind to it all right. Without the correct orientation, the reaction will not occur. And also, there will be no efficient catalysis. That's what it is meant by no reaction will occur. So, when you are having your substrates binding to the active site of the enzyme, it's like you have combined two things, right? So, we are calling it a complex. So, that's what we are referring to as the enzyme substrate complex. So, ES. So the ES complex positions the substrate and the catalytic group in the possible orientation. The moment you are having this particular enzyme substrate complex, this means that you are going to have your substrate being positioned at the correct orientation for catalysis to occur. And it occurs very rapidly. That's why you're saying that enzymes are catalysts. They speed up reactions. All what I'm slowly discussing here, it occurs at a very faster rate. It's very fast. The moment you are going to have your correct substrate binding to the active site, it changes its orientation, fit into the catalytic group, and the reaction will occur. So this, let me clean it so that I will show you the correct illustration. All right, let me choose my pen back. So this is the active site and this is the substrate. This substrate can fit directly into the active site, and you can have your enzyme substrate complex only if the catalytic groups in the active site matches with the orientation of the substrate. If not, you can have your substrate binding to the active site of the enzyme all right, but there will be no complex being formed. The substrate will just be sitting into it, no complex will be formed, no reaction will occur, you are not going to form any product. So the structure of the enzyme. Types of enzymes. Enzymes are usually classified into groups. They are classified into two main groups. We are having the simple proteins or the simple protein enzyme, and we are also having the conjugated proteins. So we are talking about a simple protein enzyme. They consist of only their polypeptide chain. They are just enzymes with their active sites, nothing else. And these enzymes are proteins. The proteins are formed from amino acid. From primary, they folded to secondary, they folded to tertiary with a correct active site formation. Their catalytic activity will also depend entirely on the protein structure. That's why we are saying that depending on how the protein is going to fold, it's going to produce a different enzyme, which is going to perform a specific function as compared to the other enzyme. There is no way you are going to have an enzyme, two enzymes catalyzing the same reaction. No, it hardly occurs. So let's move on. For the conjugated proteins, it requires a non-protein component for its activity. So that will lead us into the term called the cofactor and also the coenzymes. When you are having your enzyme, let's say this is our enzyme. Even with the active sites and everything, the enzyme can still bind to substrate all right, but no reaction will occur. The reason why no reaction will occur is that it needs a non-protein component. It needs another thing to bind to so that it will enhance the functionality of the enzyme. If this particular thing is not there, there's no way you are going to have your enzyme being functionable. So they may require a metal ion or a small organic molecule called the coenzyme. So they need either a metal ion to bind to it for it to occur, or they need a coenzyme. That is for the conjugated proteins. Simple proteins, they don't need anything. All they are composed of is their polypeptide chains made up of their active sites, and the catalytic activity will depend on only the protein structure. For the conjugated, their catalytic activity will depend on the protein structure, the active site, and also a coenzyme or a metal ion. So now enzymes are made up of one or more polypeptide chain. If you are looking at hemoglobin can be classified as an enzyme, right? Where we are having four subunits. Where are these four subunits coming from? These four subunits are polypeptide chains that have also folded to give you a different proteins that have come together. That's why we are saying that an enzyme are made up of one or more polypeptide chain. This is a typical example of a polypeptide chain. You can also have just a single enzyme made up of just one polypeptide chain that has been folded with its correct active site. So these chains fold into complex 3D dimensional shapes due to their interactions between the amino acid chains. And we talked about this in BCMU 201 when we were talking about how proteins fold. When a protein fold, they can have some interactions such as the hydrogen bonding, covalent, the metal ions. They form a lot of interactions. And these are the ones that help stabilizes the 3D dimensional shape. Sorry, the 3D shape. Okay. So the folding produces a compact globular structure with a hole. The hole is what I've been drawing always, or the cleft. On the surface of it, that's where your substrate is actually going to bind to. And that's what we are referring to as the active site. So the function of the active site is to bind to the substrate. That's one. It also contains the catalytic groups. That's two. It carries out chemical reactions. That's three. The active site is highly specific, right? So highly specific in a way that only certain substrates can bind to it. This is the active site. If you are having two things here, if this is the structure of your active site and you're having two substrates, one is round, sorry, you're having three substrates, another one is triangle, and the other one is kind of similar to the active site. This active site is specific. One, this one cannot enter, so this one is out. This one cannot also enter because of the shape. This one is also out, but this one, it can fit into it all right to give you something like this. And your catalysis can occur. Your catalysis can occur only when the catalytic groups are in the correct orientation or they matches that of the substrate for the reaction to occur. If that's not the case, you can still have this one sitting into the active site all right, but no reaction or no catalysis will occur. So the unique 3D structure of enzymes and the precise arrangement of the catalytic groups in the active site make the enzyme highly efficient and specific. So because of these two factors, the shape of the active site and the presence of the catalytic groups there, as well as the binding groups, it makes the enzyme very specific in its reaction. Meaning you can have a substrate having the same shape as the enzyme, but if it's not having, or the enzyme active site, if it is not having the same catalytic groups as that of the catalytic groups in the enzyme's active site, your reaction will not occur. That makes enzymes more specific in their reactions and also efficient. So let me clean it and show you how everything works. So let's look at this enzyme. This is the substrate and this is the active site. You can see my pointer, right? Let me use the pointer, right? So this is the active site. This is the substrate. So you see, there's something called induced fit and lock and key model. I'll be talking about it soon, but we have to focus on this particular concept. Induced fit, meaning look at the substrate. It looks like this substrate can bind to the active site, all right, but there will still be space around it. There's a model that explains that when this substrate binds to this active site, we are going to have a conformational change. This enzyme is actually going to change in shape in order to hold this substrate very well. It, it's like it changes its shape in order to grab the substrate, and that is done by the binding groups. And in the process of undergoing this conformational change, it's actually going to interact with the catalytic groups in the active site, and this will release an energy. This energy will make, or it will release an energy that will actually compensate for the high energy of the enzyme substrate complex. When we reach there, we will talk about it very well. When we get to the models of the enzyme bindings, we will look at it. We are having the lock and key model and we are having the induced phase model. So let's look at the features of the active sites. I've already discussed it earlier, but we can still talk about it again. The first sentence is just an additional information that the structure of an enzyme can be studied using a technique such as the X-ray crystallography, which provides information about the arrangement of the atoms within the enzyme and the nature of the active site. So with this, we can actually determine the nature of the active site, whether it is square, whether it is complex, whether it is circular. We can use the X-ray crystallography to check it out. The active site is a specialized region of the enzyme where one, the substrate binds to, two, catalysis take place. So what happens in the active site? Binding of the substrate to the active site. Also, catalysis also occurs there. So for a substrate to bind to an enzyme, it doesn't bind to just any part, it binds to the active site. Also, when it binds to it, that is where catalysis occur. An active site is not only complementary in shape, but also it contains functional groups that are positioned in correctly for efficient binding and catalysis. So this is what I was saying, that you can have your enzyme all right. You can have your enzyme all right with its active site, and this is our substrate. The active site is not only complementary to the shape of the substrate, as you can see. Yes, it is complementary because this one can fit into it. It also contains some functional groups. Remember when we are talking about proteins, where we are having our amino group bonded to the carbon, bonded to our COOH group. Here we are having our hydrogen, right? Let's say this one is hydrogen. This side is R group, right? R groups, usually some of them are ionizable, especially histidine, glutamate. They are all containing ionizable groups. They are functional groups. Glutamate, for instance, having the COO minus or the COOH. It's a functional group. It's a carboxylic acid group. All these things are positioned efficiently in this active sites. So they are positioned in a way that these functional groups are meant to interact with the specific groups on the substrate. So even though the substrate is having the same shape, if it is not having those groups on it, for the interaction of these functional groups in the active site to interact with the substrate, you are going to have your substrate sitting in the active site, but no reaction will occur. That is what it is saying. So, essential features of the active sites. The first part is the complementary shape. The active site has a shape that matches the substrate, like I've been saying always. and illustration is what I made earlier. So this allows the substrate to fit specifically into the active site, forming the enzyme substrate complex. The second one is the binding groups. The active site contains groups that binds the substrate through hydrogen bonding. You see, so in the active site, we are having binding groups there. The binding groups are also amino acids that are deposited in the active site. They are having hydrogen bonding. They are having some R groups in there. These R groups, some of them undergo interactions such as the hydrogen bonding, the ionic bonding interactions, even hydrophobic. For instance, valine. Valine is a typical example of the amino acid or groups that undergo hydrophobic interactions. So if you're having valine or hydrophobic side chains as binding groups, and the substrate that's also coming inside is also having hydrophobic parts, it can undergo hydrophobic interactions, holding this substrate firmly to the active site. So this interaction is the one that helps stabilize the enzyme substrate complex. When these interactions occur, so what do you think it will happen? It releases energy. And when I was talking about the energy profile, I was saying something like there's a transition stage here. This is where your enzyme complex, your enzyme substrate complexes, and it is having higher amount of energy. So what do you think it will happen if you are having higher amount of energy for a compound? It will be very unstable. You get it? It's a positive value. It's very unstable. A positive value. Now let me talk about it in the thermodynamic aspect. You are going to understand this thing very easily. This is a positive value. Remember, we are moving from zero upwards. These interactions, the hydrogen bond interactions, ionic interactions, hydrophobic interactions, and co, they occur in the binding groups of the active sites. When this thing happens, they release energy. If you are saying something is releasing energy, if you are saying you are releasing energy, what do you think is happening? It's a negative value, right? Exothermic, negative value. So already you are having your enzyme, your ES complex, having a high amount of energy, but you are also releasing energy. The energy is negative. Then, positive plus negative, you shouldn't expect your positive value to increase. It's supposed to decrease. If it is decreasing, this means that you are going to have your ES complex around this side. The energy has reduced. A compound that is having lesser amount of energy in it, it is more stable. That's why you are saying that these interactions help stabilizes the enzyme substrate complex. And the third one, catalytic groups are precisely positioned within the active site in order to assist the chemical reaction. I've been saying this thing always. If the substrate can even bind to the active site, binding groups interacting, if you are still not having your catalytic groups to be precisely positioned in the active site, you are not going to have any chemical reaction occurring. So correct orientation also increases the efficiency and the speed of the catalysis. Let's move on. So in the active site, what I've talked about here, in the active site, we have the shape matters, binding group matters, catalytic groups matters. The main parts or the main things that entails the active site is what I've said: shape, binding groups, and active sites. Sorry, the shape, the binding groups, and the catalytic groups. And under the catalytic groups, we are having the side chain amino acid. That one's I've already explained it. We'll be talking about the cofactors soon. The cofactors can be metal ions, it can be small organic molecules such as the coenzymes or the vitamins. So let's move. Catalytic groups in the active site of the enzymes. So catalysis is carried out by specific functional groups found in the amino acid chain. You get it? So in the catalytic groups, the catalytic groups are actually functional groups, right? Part of the amino acid side chains. They are functional groups. Now let's look at some examples. This is an amino acid. Serine is an amino acid. And remember that it's having a side chain having the OH group. Cysteine is having the SH group. Look at the catalytic functions. For serine, if you're having serine as a catalytic group in the active site, It functions as what? Binding of acyl groups. It binds acyl groups to another substrate, or it is capable of transferring an acyl group from one place to another. Look at cysteine, for instance. Cysteine also does the same function. Cysteine alone is not the one that's performing the function. Its functional group, its side chain. Remember, the functional group here is SH, the thioester group, or the thio group, the SH group. That is what's actually making the binding of acyl group possible. If you look at tyrosine, tyrosine is having an OH group there. The tyrosine on OH group causes hydrogen bonding to the substrate. It makes hydrogen bonding to the substrate possible. So in the active site, if there should be a hydrogen bonding and you're having tyrosine there, the OH on the tyrosine is the one that is doing that. That is the phenolic hydroxyl group. In serine, we are not having any phenolic, we are not having any phenol group there. We are not having any benzene group. We are having OH attached to the side chain of the amino acid called the serine. In tyrosine, we are having a phenolic group or a phenol group. And in this phenol group, we are having the OH. Aspartate is having the COOH. Glutamate is having the COOH. What do? This is the function that they do. They transfer protons or they enhances the cation binding. So with this, I hope you can figure out everything. And there's no need for you to actually learn all these things, learning the side chain reactiveness, the catalytic functions. No. You just have to understand the concepts that in the side chains of the amino acid acting as the catalytic groups. These are the things that they do. It's not the whole amino acid. It's rather the functional groups on the amino acid side chain. They are the ones that they are performing these catalytic functions. It's not the whole amino acid. So these groups, they are ionizable. Yes. Look at OH. OH is ionizable. It can lose its hydrogen. The thio group, it can also lose its hydrogens, ionizable. This one can also lose. This one can also lose. Even ammonia can accept a hydrogen. So proton transfer, it can take in the hydrogen or it can hold the hydrogen for the meantime and then transfer it later. So on a normal basis, let me just take one and explain it to you. How we are saying that the ammonia is, let's say you are having your NH. It's actually supposed to be NH2, right? Because the nitrogen is also bonded to another 10 else. But here in this case, you are having NH3, meaning it has taken what? Extra proton. It can hold on to this proton, and when it gets to the other substrate, then it will transfer it to give you your NH2 back. What did it do? It actually transferred the proton, or it enhances anion binding. So even with lysine, this is a typical example. The same thing as the imidazole group. They also function as the proton transfer. Look at this side. Look at this side. It can lose its hydrogen to just give you N. It can also accept a hydrogen to give you your N plus, and it can be transferred to another substrate. Now let's look at the cofactors. So metal ion cofactors. So these ionizable amino acid, or we are having ionizable amino acid, right? Yes. Of the side chains of this amino acid or the enzymes are important in the catalysis, like we've already talked about earlier. They do not catalyze all biochemical reactions by themselves. So you need a metal ion. This is because amino acid chains are generally poor electron donors. Sorry, they are literally poor electron acceptors. They don't have electrons. You get it? They are electrophiles. So many enzymes will require additional non-proteins called the cofactors to assist them in their catalysis. So importance of these metal ions. About 30% of all enzymes that have been studied, they require a metal ion for their catalysis or catalytic activities. So metal ions help enzymes by accepting the electrons. Because they are poor electron acceptors, we need a metal ion to accept these electrons for them. If they are poor electron acceptors and they also accept electrons, they become unstable, right? So the presence of these metal ions will also stabilize the charges. If they are accepting electrons, what's the charge of electron? Charge of electron is negative, and most of the metals like Mn2 plus, Mj2 plus, you see, they are having positive charges. So positive and negative, right? They tend to stabilize it. They stabilize this particular um charges and making the enzyme more stable, or it makes the catalytic activity more stable. It also assists chemical transformation during the reaction. If you are converting your substrates or your reactants to products, sometimes it assists it in the chemical transformation. If there is no metal ions there, you are going to have your enzyme to be unstable. Remember, because it's going to form this complex, your enzyme substrate complex is going to be unstable. And when these things happen, you're actually going to find your enzyme difficult in transforming this particular substrate into the product. So when this thing happens, you need your metal ions in order to stabilize these enzymes and enhance the chemical transformation during the reaction. So cofactors can be divided into small organic molecules called the coenzymes or the metal ions. And the metal ions, remember, if you're having your enzyme and it needs a metal ion, right? A metal ion can bind to it. When a metal ion binds to it, it can bind to it tightly or it can bind to it loosely. So you're having the loosely bound metal ions and you have the tightly bound metal ions. The loosely bound metal ions are called the activator ions. For them, they can be released after the reaction. But for tightly bound metal ions, for them, they are called metalloenzymes. Metalloenzymes, they are like permanently attached to the enzyme to enhance its activity. Now, let's look at the small organic molecules, the coenzymes. The coenzymes, they also have the loosely bound and the tightly bound. The loosely bound, we are referring to it as the co-substrate. The tightly bound, we are referring to it as the prosthetic groups. Prosthetic groups, they bind to the enzyme. They are not metal ions, but they bind to the enzyme very tightly. So types of the metal ion enzyme, we are having the metal enzyme, like I said. So these metal enzymes, right? They are metal ions that are tightly bound to it and permanently associated with the enzyme protein. Like I said, example is a zinc containing enzyme. So when you are having an enzyme bounded to a metal ion, and this metal ion is zinc, right? We are referring to this as the zinc containing enzyme. It tends to bind to it very, very tightly. In other cases, permanently associated with the protein. For the metal activated enzymes, these enzymes contain metal ions, but the metal ions are loosely bound. So after the catalysis or after the reaction, they tend to be removed. Now let's look at the types of cofactors. Cofactors may bind to enzymes in different ways. One is the co-substrate. So the co-substrate, they bind temporarily during the reaction and they dissociate after the reaction is complete. So co-substrate will be related to metal activated enzymes. Just that the metal activated enzymes is related to metal ions, while the co-substrate is related to organic smaller molecules. Prosthetic groups on this side, they are tightly and permanently bound to the enzyme, usually at the active site. Keep this thing in mind. Usually they are bound at the active site. That's why you are going to have your prosthetic groups. They remain attached throughout the catalysis. Even after the reaction has occurred, they are still attached to the enzyme in the active site. Now, there is a term like apoenzyme and holoenzyme. The protein portion of the enzyme without its required cofactor. So when you are having an enzyme, let's say this is your enzyme, right? I didn't even add the active side, but don't worry. And you are having your enzyme, and this enzyme is this part of the enzyme that requires a cofactor for it to work. If there is no cofactor there, then you are going to have an apple enzyme, that enzyme is non-functional, it's inactive because it requires its cofactor for it to be functional. Without the cofactor, how will it work? So apple enzyme is the name that is given to the inactive enzyme. But the moment you attach its cofactor to it, then it becomes a hollow enzyme. So a hollow enzyme is equal to your apple enzyme plus your cofactor. But your apple enzyme is the protein portion of the enzyme. There is no metal ion, there is no vitamin, there is nothing. There is no other organic molecule. So this is it. This is your apple enzyme, non-functional with your active site. But the moment you are having your coenzyme or your cofactor being bound to it, see where the cofactor is binding to. The cofactor is binding to the active site, a portion of the active site, binding to it. Now it becomes functional, so it's capable of converting substrates to products. So apple enzyme, the protein portion, inactive. The cofactor is the non-protein portion, the activator. When it binds to it, you are going to get a hollow enzyme, a whole enzyme active. Very active, it can convert your substrate to product. Now, coenzyme factors. So coenzymes are organic molecules that act as essential factors for certain enzymes. They are non-protein, like I said. It can be vitamins. Yes, it can be vitamins. Most of them are vitamins. Relatively small as compared to the enzyme protein, like you saw. You are not expected to see something like your coenzyme bigger than the active site. No. Then this is that where are they going to fit into in the active site? They should be smaller enough to fit into the active site with a space for the substrate to also bind and being converted to products. So without the required coenzyme, some of the enzymes cannot catalyze the reactions efficiently. So with your reaction that you are expecting for it to happen, it won't happen because the right coenzyme is not there, or there is no enzyme, or there's no coenzyme that is there to enhance the efficiency of this reaction. So functions of the coenzyme. Coenzymes mainly assist in transfer. Now let me try and move back to the same thing, but let me explain this one. Electrons, they transfer electrons, they transfer protons, or they transfer functional groups such as the acetyl group, a methyl group, or an amino acid. Now let's look at something here. There's something I want to show you. If you are having this is your coenzyme. Now we are having two substrates coming in for them to be converted to products, right? If this enzyme is not having its coenzyme there in the active site, and in this particular reaction, there's supposed to be a transfer of electrons from this particular substrate, let's say substrate A, to substrate B. If there is no coenzyme there to transfer electron or protons from A to B, then this means that the reaction will not be inactive, right? The reaction will not be active. You're not going to have your reaction occurring. We need this coenzyme to do that. If there is also a transfer of, let's say, an acetyl group or an amino acid from A to B, the coenzyme is the one that does it. And I'm referring to the enzymes that require their coenzymes or cofactors to work. So that explains what I've done here. Now, coenzymes can temporarily combine with substrates to form a covalent complex. So when you are having your coenzyme, right, your coenzyme can actually bind to the active site. The kind of bond that they form is the covalent transient bond or the covalent complexes. So these complexes create a highly reactive electrophilic groups that function as electron sinks. So I'll be getting time in the next few slides. I'll be talking about electron sinks, right? The electron sinks, they are groups on the molecule that holds electrons, right? Because the enzyme itself cannot hold the electrons. So they hold the electrons and help stabilizes the active site of the enzyme during catalysis. Okay. So the relationship between coenzymes and vitamins. Many coenzymes contain vitamins as part of their biological active structure. So if you're having your coenzyme, where is the pointer? Try. So if you're having your let's say if this is your coenzyme, right? Your coenzyme, some part of it is made up of vitamins that helps it or play an important role in the active structure. So vitamins are often important because they serve as a precursor for the coenzyme required in the metabolism. For you to form your coenzyme, what this slide is saying that for you to form your coenzyme, sometimes you require your vitamins. You need some vitamins to actually enable the formation of the coenzymes. So your coenzyme function, the electron transfer, right? If it functions as electron transfer, acetyl transfer, methyl transfer, or amino transfer, this is what you are doing. The group that has been transferred here is an amino group, a methyl group, that's what you are transferring, an acetyl group, or protons or H plus and electrons, that's what you are transferring. So if you are saying that a coenzyme functions as an electron transfer, this means that these are the groups that it transfers. So electron sink is actually part of the coenzyme, like I said earlier. It's a part of the coenzyme that accepts and stabilizes electrons during a reaction. It acts as a temporary storage for the site of electrons. So if you are having your enzyme with your coenzyme there, and now there's a transfer of electrons from one particular compound or one particular reactant to another reactant, the enzyme cannot hold the electrons. Rather, we are having a portion of the coenzyme called the electron sink. It's like a pocket, the one that receives the electrons, hold it, wait for the right substrate to come, and it transfers it to it. That's why you are saying that the electron sink is a part of the coenzyme that accepts, stabilizes the electrons during the reaction, and it also acts as a storage for the site of the electrons. The enzyme cannot contain the electrons, or else the enzyme will rather become unstable. And if the main thing that is making our reaction okay is unstable, then this then the reaction will not okay. So that's the function, that's the importance of the electron sink. So it stabilizes unstable reaction intermediates. It also prevents reaction from breaking down. For instance, for a reaction to break down, this means that the enzyme is highly or the enzyme is having a high amount of energy, highly unstable. It also makes electron transfer easier, and it increases the reaction speed because it's not going to make the active sites of the enzyme unstable. For that matter, you are going to have your reaction speed increasing. So without the electron sink, many biochemical reactions would be too slow or unstable to proceed efficiently. So now let's look at this table. This table represents coenzymes and their metabolic functions. So you can look at it later. You can pause the video too and also go through it. We are not actually meant to learn all these things. Yes, but there are some key things that I'll be mentioning. Coenzyme, pyridoxal phosphate, that was actually found in urea cycle and all those things. That's where you are having your transamination, decarboxylation, and isomerization. So coenzyme especially, they act as acyl carrier. So I'll take my time and make a video, and I'll also post it on my YouTube channel for you to go and watch it, on how coenzymes work as an acyl group carrier. We are also having the biocytin. We are also having your NAD plus, your nicotinamide adenine dinucleotide. We are also having our flavin adenine dinucleotide. All these things are there. So the CS, if you see CS there, like what you are seeing here, CS means co-substrate. If you see PG, PG means prosthetic groups. Co-substrate means it binds to it temporarily. After the reaction, it gets removed. But for PG, like I said earlier, tightly bound to it, and usually it is permanent. So this one too are also enzyme containing or requiring metal ions. Some of the enzymes also require metal ions. Example is the cytochrome oxidase. They require these ions, which is the Fe2 plus or Fe3 plus. Catalysts also do same. So these are also series of enzymes. Pyruvate kinase, we are having a pyruvate hexonase, we are having hexokinase, we are having the ribonucleotide reductase. All these things are enzymes with their respective metals that they require in order to make them function well. That is, they are requiring metal ions cofactors. Now, once you are ending the lessons, let's look at the structure of coenzymes. So, nicotinamide adenine dinucleotide, which is NAD+. We are having NAD plus and we are having NADP plus. Let's look at the structural difference before we move into the slides. The structural difference here, we are having NAD plus, having two phosphate group, but NAD plus is having an extra phosphate group here, and everything looks the same. Nicotinamide here, we are having an adenine here, we are having an adenine here. Everything looks the same. These are sugar, these are sugar. Just that on the second sugar of the NADP plus, on the second carbon of the sugar, we are having, which is the C2 prime, we are having a extra phosphate group, or it has been phosphorylated. So that's the difference between them. So we can move on with that. Then the main or the biological role of the NAD plus. So it mainly functions as a catabolic process. Catabolic means you are breaking down molecules, right? It also functions in energy production. These things are energy reducing equivalents. Energy reducing equivalents mean they help in the production of energy. So they can convert nutrients into ATP. If you are having NAD plus can convert nutrients into ATP. So NAD plus is actually required in some of the biochemical reactions such as glycolysis. These things will be explained earlier in intermediate metabolism. This is not the moment for us to discuss about this, but I know we've talked about electron transport chain earlier in structure and functions of biomolecules. So that is the major role of the NAD plus, which is the nicotinamide adenine dinucleotide. Now there's something I want to make here. We are having NAD plus and we are having NADH. NAD plus and NADH is the same, just that one is oxidized and NAD plus. Let me put it this way: one is oxidized nicotinamide adenine dinucleotide, and NADH is reduced nicotinamide adenine dinucleotide. The same thing applies to FAD and FADH2, the flavin adenine dinucleotide. We're having the reduced and we're having the oxidized. The one without the H2 is the oxidized, and the one with the H2, which is the FADH2, is the reduced. So NADP plus, let's look at its major roles. So it functions as anabolic process. That is building of what? Molecules. So if you are building, let's say, glucose, especially in Calvin cycle, where there's a formation of glucose, we require a lot of NADP plus. They have a lot of energy. Also, in a human system, fatty acid synthesis. When we are synthesizing fatty acid, we require a lot of the NADP plus. They produce a lot of energy and they provide the reducing power for the biosynthesis of these reactions that occurs in the living system. So these things are coenzymes, like I said earlier. The reduced form. So this is the reduced form. So reduced NADH, reduced NADPH, and this one is NADP plus, NAD plus. Now the main role is what I've said here. The main role of the NAD plus is for catabolism, and the other one is for anabolism, that is building up. Now the role of the NAD plus. So let's look at it. We are having some equations that explains NAD plus to NADH. If you don't look at it very well, you would think that, oh, we are just adding a proton to it, but that's not the case. This is because gaining electrons, for you to convert NAD plus to NADH, you need 2 electrons and you also need a proton. How do we get this proton? We get this proton by H2. If you're having H2, you need to convert it to give you 2H plus and your 2 electrons, right? making it balanced, and your 2 electrons. Now, even though it is called 2 electron reduction, NAD plus does not take both of the electrons direct. That's what you should take note. It takes it one at a time. It takes 1 electron. So the NAD plus is going to take 1 electron and is going to be converted to NAD, right? NAD. And now it accepts another hydrogen again, which is what? H plus. But bear in mind, the thing is not NADH plus, it is NADH, meaning it comes to take the extra electron or the remaining 1 electron to it to give you NADH. So it first accepts a hydride, right? The hydride is equal to 2 of the electrons plus 1 of the H plus, and the second proton is released into the solution. So this second proton, the second proton is the H plus. Remember, we are using only one of the H from the 2H plus. We are releasing only one, and we are using one. So this second proton is being released into the solution. So the conversion or the reduction of NAD plus to NADH releases protons into the solution. That is also another factor that you should keep in mind. Now let's look at it. This is a proton donor. In this case, I was using H2. Similar thing also happens here. We are having 2 of the hydrogens. Now we are going to have this nicotinamide. The nicotinamide part is actually going to accept a proton. So we are going to have a proton being added here, and this is what you are going to get. The extra hydrogen here will also be released into the solution, and you are going to have only X. So based on what I've explained earlier, you can understand this one very clearly. So this represents the total equation, where one of these proton is being released into the solution and one of the proton is being added to it. But in general, 2 electrons is being used. That's why we are referring to it as a 2-electron reduction. Let's look at coenzyme A before we end the lessons. For coenzyme A is an essential coenzyme involved in energy metabolism. Now, it is a small organic molecule. Yes, it's a small organic molecule that transfers, or it helps enzymes to transfer acetyl group. This is an acetyl group. This is a structure for acetyl group. So this coenzyme A, look at this coenzyme A. Co-A will bind to the acetyl group, and now you are going to have CoH, right? So the CoA's primary role is to act as a carrier of the acetyl group. It transfers an acetyl group. So it forms a high energy compound called the acetyl-CoA. That's the reason why we are having an acetyl-CoA to be called the high energy compound. High energy compound in a way that it can actually hydrolyze to give you back your acetyl group and your CoA. The main function of this CoA is to help in transferring this acetyl group to a different compound. So let's say we are having compound. Let's say we are having this compound and it is having a CoA here, and you want to transfer this CoA to this compound, right, which is B. We require our CoA. CoA will actually bind to this acetyl group to give you acetyl-CoA, and now it will move out and it will be transferred to this particular molecule. When it gets to this molecule and you're having your acetyl-CoA there, your CoA will hydrolyze to give you your acetyl group and also your CoA. So now we are going to look at the structure for CoA, and you understand all whatever that I've done here. Look at the structure for CoA. Let me clean it so that. Look at the structure for CoA. The whole of this is coenzyme A plus this SH group. We are calling it CoH. So coenzyme A, you can also refer to it as the CoH. It's a very huge molecule. Look at the structure of this coenzyme A with its thiol group. Now, it binds to an acetyl group to form an acetyl CoA. So the thiol group is highly reactive because of this particular part. It's highly reactive. It makes the CoA highly reactive and capable of binding to the acetyl group to form this high energy ester bond or the thioester bond. So this is what you are referring to as the thioester bond. Now it allows the transfer of the acyl group in a metabolism. So let's say in a particular reaction, we want to transfer an acyl group from one point to another. The function of the CoA is to actually transfer this acyl group in the metabolism. So now this brings us to the end of lesson 1. And the next topic that we are actually going to treat is the classifications of enzyme. So make sure you do well. Before we talk about the classifications of enzymes, I'll be posting some questions inside, and I urge each and everyone to complete this lesson, understand this very well. If you have any problem in any part, you can just type it in the comment section, or you can type it straight in the group so that I reply to you with immediate effect before you start solving the questions. So that brings us to the end, and thank you for your time and also for watching the video. See you all on the next lessons on classifications of enzymes. Bye.